let's go. Hi, my name is Amanda. I'm an undergraduate student, and this is a bit of the project I've started developing during the last week in this course. So, my project is about neocortical microcircuit modeling employing Potsdam-Diesmann network model and multi-compartmental neurons. So, what that means. And when we talk about neocortical, it means that the brain structure that I want to model is the neocortex. It is considered the most complex brain structure, and it's responsible for the most cognitive functions. It consists in 6-layer arrangement. When we say microcircuit, we think in the concept of canonic circuits that can be seen like building blocks supporting brain function. In general, when we say canonical cortical microcircuit, it means general laminar connective scheme used through all neocortex. So, okay, I would like to model a neocortical microcircuit, but with what network model? Using Potsdam-Diesmann. This model is a data-driven network architecture, and when we talk about the simple neuron points, so in terms of reproducing the canonical cortical microcircuit connectivity using the simple biomorphological detailment, it is the most complete and detailed model. So what Potsdam-Diesmann tells us? It is a model of 8,000 neurons. Opa. Just a second. I think I have a delay on my screen, but okay. The slide is not passing. I don't know what I can do.
Yeah, it's still on the same slide.
To fix it. Yeah, it's still on the same slide. That's a problem.
Try to close it and restart it?
Maybe I will try. I'm using the camera and other computers, so maybe internet, I don't know.
Yeah.
I will shut down the transmission. I will try to load it again.
Sounds good.
So that's strange because it is still showing. Should it stop? Opa. Oh, that's bad. I don't know what to do. My internet was just working fine till now.
Let's see. Well, yeah, you'll be the last presentation before lunch, so you can take as long as you need. If you need to restart your computer or Chrome or something real quick, go ahead and do that.
Okay, I'm sorry.
Oh, no worries. We understand.
No worries at all, Amanda.
Yeah, so maybe a couple minutes, guys, if you need to get a drink or something, feel free.
I'm sorry.
No worries.
I will
just
try to
maybe
reopen it, and I don't know. What can I do?
This is Google Meet's fault. Amanda, if you want to try and send me your slides to me and I can try to present them if you want.
Oh, I think that's a good idea because here in my camera I have internet and then in my computer I don't. So I
have here. I think I can present. Sorry.
Okay.
I have.
Oh, just. Okay. Oh, Fernando, I'm seeing your screen, right?
Yeah.
Oh.
Sorry. Yeah, I think you stopped.
Yeah.
Here.
So can you hear a click?
Sorry?
Fernando, can you hear a click?
Yes.
Okay, so when the click, you you go on, okay?
Pavlovian training. I love it.
Yeah, very automatically.
Okay. So I'm
It's
sorry,
conditioning
just
stimulus.
I'm sorry, just getting back. I'd like to network model. What is Potsdam-Diesman says? It is a network architecture. It is data-driven, so it's based on experimental data. And in terms of when we talk about the simplest point neurons like this, the lowest morphological detailment, like integrated fire neurons, it is the most complete in detail models in terms of reproducing the canonical cortical microcircuit. So it's a good starting point. What Potsdam-Diesman tells us? It is a network that consists in 8,000 leaf neurons, and it's nice because it has a scale of 1 neuron in the simulation, 1 neuron in real life, and supposedly, and it is consisting in 4 layers, 8 population, 1 excitatory and 1 inhibitor for each layer, and its experimental connection probabilities are very detailed. So the last but not least, multi-compartmental models, multi-compartmental neurons are spatially distributed neurons, and they allow us to improve the biophysical detail of our model. So this is a thing that Potsdam-Diesman model don't have, so it would be a step further. So what is my project background? And we have as a starting point the NetPyNE implementation scale in Potsdam-Diesman cortical microcircuit model, a paper that is still in the oven with Cecilia Romara et al. And in the paper, they re-implemented the model, the Potsdam-Diesman, using NetPyNE, the tool we want to learn about it. And they added also a scaling method, so we could simulate the network maintaining its firing rates and all statistics with a small number of neurons, what can be good working like in personal computers. And what is interesting here is the proof of concept they introduced. That was the implementation of a multi-compartment neuron in this model. So the leaf neurons were replaced by 6 compartment pyramidal neurons with a lot of conductance-based channels. So here, what was the result of this re-implementation and this change in the neurons? The simulation of the new biophysical detail in the scale of 1%, so the rescale method was used, was able to obtain physiological-like activity in all populations. So it's a good start because we're able to put a more detailed model that allows, for example, LFP measurements that can be after measuring the lab to validate our model. So it's a great thing. It's important. So what are the motivation? Why neocortex? Because we have challenging questions like how can one network produce different patterns? So collective activity different, and that's interesting to investigate. Why do we work with Potsdam-Diesman and try different neural models? Because maybe I think it's interesting to investigate what is the level of modeling that is necessary to be able to provide simulation results that are consistent with what we have experimentally. And the last but not the least, my major motivation during doing this was to be able to play with NetPyNE to see how it works. But I never used this tool, so I think it was the major motivation for me. So project implementation. The idea was to use the Potsdam-Diesman network model concerning getting the core its core, that is the cell type specific connectome probabilities matrix. So it's the most important things in the Potsdam-Diesman network. Using multi-compartmental neurons and the network scale that allowed me to run my computer, for example. So the interesting thing is that to apply the multi-compartmental neurons, different from the Cecilia et al., we tried to use different neuron cell types for each layer. So these cell types came from the area S1 from rat, from Macron results. So, okay. So we are going to implement Poisson dismal with compartmental models of this specific area. So NetPyNE requires spatial dimensions. So we used the cylinder format to the column of the microcircuit using the dimensions and layer thickness and boundaries from the area assumed from rect. And an important thing to do after LFP measures and other further stuff is to maintain the densities like in the real red area. So we did it. And to get the cell types, we had with Macron data 55 layer-specific morphological options. So in this first idea of implementation, we did a selection. The idea was to select one cell type to represent the whole population. So how we did it? We looked for the cell type's composition per layer. So we can see, like, for example, for the excitatory neurons, we have just one or two, but always a main neuron that is present in the major quantity. But the other side, for the inhibitory neurons, we see that we have a great diversity. So we try to get a bit of this diversity in our implementation. So in the end, what we get it for excitatory populations, all pyramidal cells, but there are 4 different kinds of cell types. And for the inhibitory populations, we get 4 inhibitory populations with diversity. So each one has actually 2 subpops, one for nest basket cells and martinet cells. So, okay. This was the first time I was simulating a network. So the first thing I wanted to see was, did NetPy created my populations in positions, in the right positions in the column? So we can see here the X view, so the cylinder from the front, like each layer. So we can see, yes, the populations and the cells are there. And we can see in the side that it is really a geometrical format of a cylinder. I think it was interesting to see, did NetPyNE created my connectivity model? So we can see here the connections, the synapses. We see that in the layers 2, 3, the layers 2 and 4, we have a lot of connections intra-layer, and we can see also the connections between the layers 5, 6, and we can also see the top view again with the connections, visualization of the connections. To see, is this model reproducing, is these connections going to where they were supposed to be? So we plotted these, and we can see here that we got a result that is very similar to what Cecilia Romano et al. Got it. So apparently the network, its connectivity, it's fine, it's okay. And the next thing was to see the morphology. Like, now we have not a point neuron, so what it's like, what does it look like? So this is, you can see 12 neurons, one from each cell population and the sub-inhibitory populations. And we can see that, yes, we have a spatial distribution and different morphologies from different neurons. Here we don't have the complete neuron, is a reduced number of compartments, but we can have an idea how things are different. And we also simulated with more compartments, so we can see the column is really getting morphology and spatial, and it's different from in its complement. So the last thing is, okay, we have a network, we have the connection. So did it was active? Did we see spikes? And the answer was yes. When we used a scale of 2%. We see that the firing rates are compatible to the Poisson-Dismal model, but for all populations except in the layer 5 for the excitatory population. We don't know exactly why, but maybe this comes from the cell type we choose to represent it. So, yeah, it's a nice first result. And we also plotted the potential traces for each cell from each population to population. So we had firings. We can see what NetPyNE can do and what it can give us. And the last thing was we plot the traces, the potential traces from, again, each one cell from each population to populations. And we can see here in the, I don't have the pointer, but in the blue one, we can see that it doesn't spike in the middle. It corresponds to the 5 inhibitory layer that was strange in the other result. So this is it. I know it's just baby steps, but I was very happy to be able to learn new things. I did never work with NetPyNE, or this is my first neuron NetPyNE and network model implementation. So I'm very happy just to see plots, just to have plots, and that's it. I know it's a long way. And I love it to watch all other presentations. I'm sorry for the technical problems, and I was very nervous, but thank you.
Thank you.
Really good.
All right, questions?
So Amanda, great job. I want to ask, so what do you think are the advantages of having these multi-compartment neurons? What do you think are some things that we can investigate now and we couldn't before with the point neurons?
I guess now with the adding of multi-compartments, it means that we have morphology. So before with point neurons, you only had the potential in one place, and now we can see how in the network, sorry, let me just describe. We can see now with morphology, we now have the potential being distributed to a path. So we can see things like we have a stimulus here, not be the same here, because we had the long path, so we can measure things like the local field potentials. In the soma will be different from the dentary, so we can see these new stuff, these kind of measures that we weren't able using single point neurons.
Excellent. Yeah.
Yep,
Totally
absolutely.
agree.
Yeah, like those apical tufts, using a point neuron for some of these enormous morphologies, it has to travel all the way from the apical tuft down to the soma, you should really probably include that in your model.